Hello guys, welcome to my channel. Today we are studying about the activated carriers and biosynthesis. So we'll just write it down here. So we are studying about activated carriers, biosynthesis. All right, so if you know, there are like two types of chemical reaction in the cell. So one is energy favorable chemical reaction and other is energetically unfavorable chemical reaction. So what does that mean? So energy favorable reaction means, so I'll write it down here. So energy favorable reaction. Right, so what is an energetically favorable reaction? So the energetically favorable reaction means, so the food molecules that we eat, so there is an oxidation of food molecule, some chemical reaction in that food molecule, and during that oxidation of food molecule, energy is released. And that energy, so that reaction that happened during the oxidation of food molecule, so that releases an energy. So that reaction would be called as an energetically favorable reaction because that releases an energy, and the energy that is released by that reaction, the energetically favorable reaction, so that must be stored temporarily because it can be moved or used by the cell to fuel unenergetically energetically unfavorable reaction. Sorry. So the energy, so when there is an oxidation of, suppose oxidation, like I'll write it here. So oxidation of food molecules, so that will give energy will be released from the oxidation of food molecule. So the energy will be first stored temporarily in some part of the cell, and like when the cell need that energy, so this energy will be used for energetically unfavorable reaction. So that will be used for the energetically unfavorable reaction, like such as the synthesis of all the other molecules needed by the cell. Okay, so and in most of the cases, so the energy is stored as a chemical energy or a chemical bond energy in a set of activated carriers. So like where this energy is stored, so it is stored in an activated carriers in the cell. Small organic molecules that contain one or more energy rich covalent bonds. So what are the activated carriers? So that are like a small organic molecules, so that contains one or more energy rich covalent bonds. So these molecules diffuse rapidly and carry their bond energy from the sites of the energy generation, so from where it is generated to the site where the energy is used for the biosynthesis or for other energy requiring cell activities. All right. So the activated carriers store energy in an easily exchangeable form. So these activated carriers store the energy that is captured from here in like an easily exchangeable form, either as a readily transferable chemical group or as a readily transferable high energy electrons. So either it's stored as a chemical group or stored as an readily transferable electron or a high energy electrons. So they can serve as a dual role as a source of both energy and chemical groups for the biosynthetic reactions. Because of that, like it can store on both the sides, so it can serve as a dual role as a source of both energy and chemical groups for biosynthetic reaction. Because for some biosynthetic reactions, they might need a chemical group as well instead of a source of energy. So this is how the activated carriers store the energy that are released by the oxidation of food molecules. So the most important activated carriers. So which are the most important activated carriers? So the most important activated carriers are, okay, so what is done? Look down. So the first one, so first is ATP, adenosine 5-triphosphate. Alright, so the most important activated carriers in the cell is, number one is ATP, and there are two other molecules that are closely related to this. So they are NADH, and the second is NADPH. So the full form of this NADH and NADPH is, I will write it down. So nicotinamide adenine dinucleotide, and for this is also nicotinamide adenine dinucleotide, and there will be phosphate because it has a phosphate group as well for the NADPH. Right, so this ATP, NADH and NADPH, so these are the activated carriers in the cell, and these activated carriers are used like a money to pay for the energetically unfavorable reaction that otherwise would not take place. So if there is no activated carriers, the energetically unfavorable reaction, that is like biosynthesis of some kind of proteins or any other molecules, it won't take place if there is no activated carriers. So the activated carriers are really important in the cell. So when a food molecule such as glucose is oxidized in a cell, enzyme catalyzed reaction ensure that a large part of free energy that is released because of the oxidation of glucose is being stored or captured in a chemically useful form rather than being released wastefully as a heat. So like for example, oxidizing sugar in a cell allows you to power metabolic reactions, whereas if you burn a chocolate bar on the street, it will just give you nothing. So there is no metabolically useful energy. So the energy that is released is just wasted. So in cells, energy capture is achieved by means of coupled reaction, in which energetically favorable reaction is used to drive an energetically unfavorable one that produces an activated carriers or some other useful molecule. So what does that mean? So I will explain that. Alright, so to explain this coupled reaction, we'll use the mechanical analogy. So for example, I'll just make a small thing here. So for example, this is a cliff, and if there is a rock falling, so rocks are falling from the cliff, right? And this is the surface, and here is the water, for example. So as per this mechanical analogy, so for example, the energetically favorable reaction or a chemical reaction is represented by the falling of the rocks from the cliff. So that means the kinetic energy of falling rocks would normally be entirely wasted. So these rocks are falling, so it has a kinetic energy, and when this hit the ground, so it will generate the kinetic energy will be transferred to the heat energy. So but when it hits ground, so the heat energy is generated, but it's totally wasted. So kinetic energy generated by the friction, that heat energy is totally wasted when the rock hits the ground. So by careful, suppose if we design something like this, careful design, okay, something like that. Yeah, I hope you can understand that. Right, so suppose if we design something and do like this, so part of this energy from the kinetic energy that the rocks are falling, we shift to drive a paddle. Suppose this is a paddle, for example, and when the rock hit this paddle, it revolves, and the rope that is tied here to the bucket, so the bucket is used to just lift the water from here down here and then this, right? So just to explain that, so the part of this energy could be used to drive a paddle wheel that lifts the bucket of the water. So now the kinetic energy is used here instead of just falling directly into the ground. So because the rocks can now reach the ground only after moving the paddle, so the velocity of the rock is less. And so that means we say that the energetically favorable reaction of rock falling has been directly coupled to the part of the energy is used to do work in the paddle. The rock hit the ground with the less velocity then, and correspondingly less energy is wasted as the heat. So energy is saved in an elevated bucket of water that can be used to do useful work. So the analogous process occur, the same analogous process occur in the cell, where instead of this paddle, enzyme play the role of paddle. All right, so the enzyme couple an energetically favorable reaction such as oxidation of foodstuff to an energetically unfavorable reaction such as generation of an activated carrier. So as a result, the amount of heat that is released by the oxidation reaction is reduced exactly the same amount, or the amount of energy that is stored in the energy rich covalent bonds of the activated carriers. So that saved energy can be used to power chemical reaction elsewhere in the cell. So this is how it works. So the most used activated carrier is an ATP. We are studying about the activated carriers. We previously studied about the ATP, and now we'll study about the NADH and NADH and NADPH. Alright, so nicotinamide, adenine, dinucleotide, and nicotinamide, adenine, dinucleotide, phosphate. Alright, so these are the two other important activated carriers in oxidation reduction reactions, and they are commonly part of a coupled reaction as well in the cell. So these activated carriers are specialized to carry both high energy electrons and hydrogen atoms as well. So that's their specialty. So the most important of these electrons carriers are NADH and the closely related molecule, that is this NADPH. So both NADH and the NADPH carry energy in the form of two high energy electron plus a proton, which together form an hydride ion. So how they carry their energy in a two high energy electrons plus a proton, which together forms an hydride ion. So when these activated carriers pass their energy in the form of hydride ion to a different molecule, they become oxidized to form. So when they pass energy, so they will become oxidized to form NAD plus, that's because they are oxidized, and NADP plus. This is how. So like ATP, NADPH is an activated carriers that participates in many important biosynthetic reactions that would otherwise be energetically unfavorable. So what this do? So like you will see a picture on your screen. So during this special set of energy yielding catabolic reaction, as you are seeing in your picture, so a hydride ion is removed from the substrate molecule and added to a nicotinamide ring of NADP plus. So this is on the left side that you are seeing. So this is how it shows. So this is, for example, that's an oxidation of a molecule, and there is an NADP plus that's there, and like it is going through that reaction and it will form NADPH because as it is receiving the hydride ion, and so that means hydrogen to form NADPH. So this is typically an oxidation reduction reaction. So the substrate that is you're seeing on the left is oxidized and NADPH P plus or NADP plus is reduced. So if you remember oxidation and reduction, so what is oxidation? Is removal of electron. That's an oxidation, and reduction is addition of an electron. So that means, as you can see on your left side, so the substrate that has been oxidized, the oxidized form of that carrier molecule and the NADP plus then receive one hydrogen atom plus an electron. So while the proton, that the H plus that has been received by NADP plus is released in the solution. Because the NADPH holds its hydride ion in a high energy linkage, the ion can easily be transferred to other molecule, as you can see on the right side of your screen. All right, so this is how the cycle is completed. So the hydrogen ion carried by an NADPH is given a readily in a subsequent oxidation-reduction reaction, because the ring can achieve a more stable arrangement of electrons without it. So that is why it is readily givable. In the subsequent reaction, which generates NADP plus and NADPH becomes oxidized and the substrate become reduced, thus completing the NADPH cycle. So this is how it goes. So the NADPH is efficient at donating its hydride ion to other molecules for the same reason that ATP is readily transferable, that transfers of phosphate. So that is the PI, right? That the inorganic phosphate. In both cases, the transfer is accompanied by the large negative free energy change. All right, so there is one more picture on your screen. So this is an example that shows how the NADPH is used in the biosynthesis. This picture is showing the biosynthetic route of the cholesterol, and this is the final stage. So when this 7-dehydroxy cholesterol comes with the at the end stage, and when the NADPH reacts with it, so that NADPH gives its hydride ion to this 7-dehydroxycholesterol to form cholesterol. So as you can see in the picture, that gives H plus and it will become NADP plus. All right, and that reaction will finish it by a cholesterol, by forming a cholesterol. So this is how it is used. So like there is a long chain of metabolic pathways, and like the ATP or NADP plus is used like this way. Alright, so what is the difference between the NADPH and the NADH? So that's nicotinamide adenine dinucleotide phosphate and nicotinamide adenine dinucleotide. So what's the difference? That's the difference of just a phosphate, a single phosphate group. So as you can see in this picture, so that will show you. So that shows the diagram of NADP plus and NADPH. All right, and there is a phosphate group at the end. So that's the difference. The NADH does not have that phosphate group. So that's the difference. So, and that phosphate group is located far from the region involved in the electron transfer in the NADPH. So as you can see in the picture, the electron transfer occurs on the first stage of that first stage of the region of the NADPH, right? Although this phosphate group has no effect on the electron transfer properties of NADPH compared with the NADH, but it is crucial for that distinctive role, as it gives NADPH a slightly different shape from NADH. So this subtle difference in conformation makes it possible for two carriers to bind as a substrate to different sets of enzymes. So that is a difference, that it can bind to different set of enzyme and thereby deliver electrons in the form of hydride ion to different target molecules. That is one of the difference. All right, so then it's like a division of labor. So like, why should there be a division of labor? The answer is in the need to regulate the two sets of electron transfer reactions independently. So NADPH operates chiefly with enzymes that catalyzes anabolic reaction, supplying the high energy electron needed to synthesize energy rich biological molecules, while on contrast NADH has a special role as an intermediate in the catabolic system of reaction that generate ATP through oxidation of food molecules. So the genesis of NADH from NAD plus and that of the NADPH from the NADP plus occurs by different pathways, so that are independently regulated so that the cell can adjust the supply of electrons for these two contrasting purposes. Inside the cell, the ratio of NAD plus to NADH is kept high, whereas the ratio of NADP plus to NADPH is kept low. So this arrangement provides plenty of plus to act as an oxidizing agent, and plenty of NADPH to act as an reducing agent, as required for the special roles in catabolism and anabolism respectively. So this is the different roles in the cell. So beside this ATP and NADPH and NADP, there are some other activated carriers are used as well in the cells as well. So like as we write, so ATP, that is the group, will just give the group that are carried for the high energy linkage, group carried. So ATP, the group is phosphate. All right, there is the common one that we studied. Here, there is one more that comes in this, that is this, and this is the FADH2, and that is, as we study, electrons and hydrogen. There is acetyl coenzyme that you will study further, so that carries the acetyl group for the high energy linkage. There is carboxylated group or carboxylated biotin, rather, that carries the carboxyl group. Right, so these are the activated carriers on this side. So there is one more than adenosylmethionine. This is a long name that carries the methyl group, and there is one more that is uridine diphosphate group. Diphosphate group that carries the glucose. So these are the activated carriers, and these are the groups carried in high energy linkage form. All right, so these are others as well, but the most important are others that we studied, but we will study more that in details when we'll study biochemistry. All right, guys, so that was just a basic of how the cell in cell the energy is generated, how it is carried, and how it is used. Right, so we'll see you in the next video. So most probably I think we'll start with the protein structure and function. So before we go deep into the cell, we'll just study this molecules and then we'll dive into the cell. All right, thank you guys for watching, and thank you for subscribing to the channel. Thank you.